Started. Welcome back to another TPD seminar. It's my great pleasure to introduce our first speakers for the first seminar today. The first is Stephan DeCarlo. He's a graduate student and Professor David Liu's lab at the Broad Institute. He received his bachelor's degree from Odeon College and worked on antibody discovery and strain engineering prior to attending Harvard. His research interests include using phage-assisted continuous evolution to engineer proteins with novel functionalities. And together with Stephan, we had Shourya, who is a research fellow in the Fisher Lab here at Dana-Farber and Harvard Medical School. He works on developing new chemically inducible degradation tags using computational methods and characterizing them using structural and biophysical techniques. He received the Cancer Research Institute Irvine postdoc fellowship for exploring the regulation of engineered cell therapies with Degron tags. And previously, he completed his PhD at Johns Hopkins University in Jeffrey Gray's group, where he developed protein docking programs for Rosetta modeling suite. And I will hand it over to you guys to tell us about your work.
Awesome. Thank you so much for the introduction. Let's get our screen set up. All right, yeah, so as Catherine mentioned, today we'll be presenting on our recently published work centered around the continuous evolution of these compact Degron tags that are regulated by selective cereblon molecular glues. And so my name is Stephan DeCarlo. I'm a graduate student in David Liu's lab. I also work very closely with Jaron Mercer, who's a postdoc in David's lab and also led all the work that we're about to present to you.
And I'm Shourya Burman. I'm a research fellow in Eric Fisher's lab at Dana-Farber. And today we'll talk to you about the story on the engineering of a new Degron system. So the ubiquitin proteasome system, this is one of the major pathways of protein degradation in the cell. And this consists of a cascade of 3 different enzymes, E1, E2, and E3. Here, the E3 acts as a platform that brings together the E2 that's charged with ubiquitin in close proximity with the substrate, which then gets ubiquitinated and subsequently degraded. Here we are showing you one of the workhorses of targeted protein degradation, and that's the Cullen ring ligase 4 system, with cereblon acting as the substrate receptor. Over the last 15 years or so, we've known that cereblon can be targeted with small molecules, and these small molecules bind cereblon and act as degraders by remodeling the surface of cereblon and allowing it to recruit certain non-endogenous substrates. And these new substrates are then ubiquitinated and degraded just like any other substrate would be. So while most of the field is concentrating on developing new degrader molecules to bring together new targets or looking for new E3 ligases to expand the target scope, what we asked ourselves was, if we had a compound that was very clean, could we engineer a new substrate to bind to it? And the reason this is useful is because it's been shown that these kind of new substrates can act as degradation tags, which can be fused to your protein of interest. So here we are presenting a classic example of the DTAG system, where FKBP mutant is this new substrate. And the molecule that triggers the degradation is called DTAG13. And when this is attached to any of these proteins of interest, such as HTAC1 or MYC, etc., this leads to rapid degradation of these proteins in an hour or two with treatment with DTAG13. Also, because this is small molecule controlled, it's rapidly reversible just by washing out the small molecule. And all of this biology can also be observed in an in vivo setting. More recently, in the labs of Ben Ebert and Marcella Maus, it was shown that this kind of technology can also be used to regulate CAR T cell therapy. Specifically, what they did was they took this IKZF3 zinc finger and fused it to a CAR in the CAR T cell setting. And with lenalidomide, they were able to regulate the activity of the CAR. And this leads to higher safety and efficacy profiles for these CAR T cell therapies. So although these technologies are quite promising, they still have certain limitations. One big limitation is that many of these degradation tags are rather large. And that presents a problem when we do precision genome editing, like prime editing, because this leads to lower efficiencies of integration, especially in a genomic setting. With some of the smaller tags, like the 8 tag, you require an exogenous expression of E3 ligase. And with the tag we saw in the previous slide, which was the IKZF3 zinc finger Degron, although this is quite small, it's triggered by these molecules like pomalidomide, lenalidomide, and thalidomide, all of which have several off targets, as has been reported by us and several others in literature. So these ligand properties are not really favorable because they either require large bifunctionals, which also have hook effects, or they lack orthogonality because they target all of these natural zinc fingers. So very recently in Amit Chaudhary's lab, there was this effort to find an imide compound or an imide derivative which lacks any of these zinc finger neosubstrates. So they took pomalidomide, and at the 4th position, they removed this amino group, and at the 5th position, inserted this morpholine ring. And they were able to show in that paper that for 14 common neosubstrates, this does not lead to any degradation. When we tried to characterize this in our study done by Catherine here is global proteomics, we can see that on multiple cell lines with pomalidomide, we saw degradation of your classical zinc finger transcription factor type proteins, but we see no degradation with PT179. Looking at longer time scale studies with Peter Chen, in RNA-seq, we see that with pomalidomide, the transcriptome is quite disturbed, and this can possibly be explained by the fact that many of the things that get degraded are transcription factors. But with PT179, there are only 3 differentially expressed genes. So now we had a very clean compound, and now Stephan will tell you how we evolved a Degron to bind to it.
Yeah, so we turned to directed evolution. In particular, the system that we used is phage-assisted continuous evolution, or PACE, which is a directed evolution system that was developed by the Liu lab. And so the way that PACE works is it basically relies on the M13 bacteriophage life cycle, whereby we can basically substitute an essential gene called gene 3, which encodes this P3 protein that's required for infection. We could substitute that for an evolving protein of interest. In this case, you can imagine it being the Degron, and we fuse it to this omega subunit of RNA polymerase, which I'll explain the purpose of that in a minute. But these phage can basically infect host recipient cells, and these host recipient cells harbor this accessory plasmid, or AP. And the AP encodes a synthetic biology circuit that the evolving protein must function and perform on in order to generate gene 3 and replicate. And so in the context of MG-PACE, we basically have this bait protein, in the case cereblon, and we can fuse it to a DNA binding domain and place its cognate operator upstream of this heavily previously reported optimized promoter that regulates gene 3 transcription. We could just physically substitute in the molecular glue by small molecule addition into the medium. And then if the evolving Degron is capable of forming a ternary complex with molecule-bound cereblon, it anchors down this omega subunit, thus recruiting RNA polymerase and promoting gene 3 transcription and translation. The host cells also harbor this mutagenesis plasmid, which encodes these mutagenic genes, which can introduce new mutations into the evolving Degron with each infection cycle. And so throughout this continuous process, we also have a constant outflow to remove those uninfectious phage, as well as dead and old host cells. And then we also have a constant inflow to provide fresh host cells for subsequent infection events. And so we initially validated the circuit just using full-length cereblon and also just the essential thalidomide binding domain, or C-terminal domain of cereblon, with pomalidomide and this super Degron, which is a chimera of IKZF1 and ZFP91, previously reported by the Ebert and Mouse labs. We substituted gene 3 for LuxAB to enable just a plate-based luminescence readout. And we're showing that data in the bottom right, where you can see nice transcriptional activation following pomalidomide administration for both constructs tested. Given the higher signal with the C-terminal domain cereblon, we decided to start evolution with this because typically higher signal indicates an easier path for evolution. And initially we started with PT179, but we figured it was too stringent because we struggled to find any solutions. And so we hypothesized that potentially we could use alternative imide derivatives that still bear phthalimide ring substitutions, but they have varying degrees of neosubstrate recruitment propensities. And so that's what we're showing here. We took a panel of a bunch of these compounds that were previously synthesized by the Chaudhary lab, and Jaron ran this beautiful parallelized PANTS, which is a non-continuous form of PACE. And you can see that by the x-axis, where we basically perform these serial passages. Throughout this process, we then track the phage titers, which is indicated on the y-axis, and typically high titers that are stable indicate that the phage have solved the circuit and basically found solutions. And you can see that in the case of molecules like 8045, where in the late passages, we start to see stabilization of those titers at high levels. And so we characterize some of those variants like SD8 and SD12. And what you can see relative to SD0, the starting point is we see nice improvement in circuit activation with their corresponding evolved stepping stones. And we also begin to see a blip in activity with PT179, at least at high concentrations, indicating that the stepping stone approach did indeed work in generating Degrons that can tolerate PT179. And so we carried forward SD12 in particular for subsequent rounds of PACE with PT179, and we're just showing that here with mutations relative to SD0 highlighted in blue. And what you can see over the course of the evolution in our transcriptional activation assay is we see a nice steady improvement with subsequent rounds of evolution. And essentially, we ended with SD20, which is shown here, which has nice, strong transcriptional activation in response to PT179. Unfortunately, though, we then turned to this previously reported fluorescent-based reporter assay to characterize ST20 in 293 T cells, and we saw that ST20 exhibited little to no improvement relative to ST0 as a PT179 responsive Degron tag. And so recall that this entire evolution was performed on the C-terminal domain only of cereblon, and based on the data I'm about to show you, we figured that potentially the N-terminal domain is kind of essential to include for evolution. So this is a previously reported paper or study that basically solved the co-crystal structure of IKZF1 bound to pomalidomide and cereblon. And you can see that cereblon's in this open conformation where the N-terminal domain is distal from the C-terminal domain. Within this same study, though, they then performed molecular dynamics and actually found that the C-terminal portion of IKZF1 was predicted to make contacts with cereblon N-terminal domain when cereblon adopted this closed conformation. And some of those predicted contact residues were subsequently mutated, and I'm just highlighting those corresponding residues up here. And you can see that they then purified those variants and just performed in vitro binding assays with TR-FRET and saw a rightward shift in the binding curves when these residues were mutated, indicating that it was impairing productive ternary complex formation. And so in the absence of the N-terminal domain, we figured that this allowed ST20 to pick up this hypermutation in the C-terminal region. And then in mammalian cells, when we're simply relying on full-length cereblon for hijacking UPS, we're basically getting unproductive contacts that is impairing the ternary complex to form. And surely enough, we performed some reversion analysis to at least get at some of what we were hypothesizing. And you can see in variants like SD20.1, where we just revert the most C-terminal mutations within SD20, we do see a leftward shift in the binding curve in the full-length cereblon circuit, indicating that potentially we're rescuing some of these previously existent contacts with the N-terminal domain. And so at this point, Jaren and I were like, all right, we just need to go back and start with some early variants and reperform this evolution again with full-length cereblon instead. And that's what we're showing here, where we performed some rounds of PANTS as well as several hours of PACE. And ultimately, this afforded SD35, which contains 7 mutations relative to SD0. And we see now in our transcriptional activation circuit with full-length cereblon that we have an EC50 value that's sub-micromolar. So throughout this entire process, though, we were basically modulating stringency by weakening the RBS strength, the ribosome binding site strength that regulates G3 translation. And so we figured that maybe we could lower it even further from this current point indicated here, but we ended up just getting phage washout. And so we were trying to figure out ways that we can alternatively modulate stringency. And immediately we turned to the idea of potentially lowering the small molecule concentration. But by doing that, you're also increasing the fraction of unoccupied apocereblon. And so we were really worried about evolving molecule-independent activity. And so in order to overcome that, we basically developed this dual positive and negative selection circuit. And so currently, we were just belonging in this regime, shown here on the bottom left, where we have wild-type full-length cereblon fused to this DNA binding domain derived from the 434 phage repressor with its cognate operator placed upstream of gene 3. We then developed this alternative system where we can take this previously reported double mutant of cereblon that's incapable of binding IMiDs. And we fused an operator pair and placed it upstream of gene 3 neg, which is a dominant negative form of gene 3. So if the evolving Degron happens to bind apocereblon, that will trigger gene 3 neg transcription and translation and poison phage replication. And surely enough, we validated the circuit and showed that they basically have superimposable sigmoidal curves, indicating that the DNA binding domain and operator pair are essentially identical in function. And we also show that they are orthogonal, whereby when we basically have a non-cognate operator and DNA binding domain pair, we don't see any pomalidomide dependent signal. And so we utilized this dual positive and negative selection circuit for the final round of evolution, shown here, where we dropped the small molecule concentration 20-fold to 500 nanomolar. And this picked up an additional mutation and identified our final variant, ST36, which now has an EC50 value of roughly 200 nanomolar. And so we then took these evolved Degrons and went back to that fluorescent-based reporter assay and characterized them. And what you can appreciate is that over the course of evolution, at least in the variants that we characterize, we see a nice steady improvement in the potency of these Degrons. We then were hoping to find a minimal Degron to also further make genomic knock-in even more efficient. And so we performed successive rounds of trimming on SD36. And ultimately, what we were able to find is that basically just the 36 amino acid span of between the first and last mutated residues in ST36 was sufficient to confer a PT179 responsive Degron. And that's shown here with ST40, which is our final human cereblon Degron that has nanomolar potencies with PT179. We further characterized ST40 as an N and C terminal Degron across a variety of different proteins. We just generated these lines using lentivirus. And following overnight treatment with PT179, we see nice dose-dependent depletion of these proteins. Furthermore, we performed a time course experiment where we see rapid onset within 10 minutes of administration, and then we essentially see no protein remaining following an hour of PT179 addition. And then finally, given ST40's small size, we decided we should definitely evaluate how efficient it is at genomically tagging endogenous proteins. And so that's what we're showing here with prime editing, which was a genome editing technology developed by the Lu Lab. We tested PLK1 as a C-terminal tagging and also BRD4 as an N-terminal tagging approach. And we can see highly efficient and very clean tagging of these endogenous genes. In the case of PLK1, we're getting efficiencies upwards of 25%. We then were able to isolate these homozygous lines quite easily. And we're then showing over here that we can degrade these endogenously tagged proteins following PT179 administration. And then finally, we just wanted to confirm that MG-PACE is actually capable of evolving a tighter ternary complex with cereblon and PT179. And so that's what we're showing here. We have ST0 in BLI, and we see a nice high affinity interaction with pomalidomide and cereblon, but we don't see any measurable affinity in the concentrations tested for PT179 and cereblon. Conversely, though, we see that ST40 binds both complexes with a KD value around 130 nanomolar.
We corroborated these in vitro results with TR-FRET, where we took a FRET donor and attached it to the Degron. Here, the Degron could be SD0 or SD40, and we took the FRET acceptor and attached it to cereblon. And we pretty much see the same result, where cereblon is able to recruit SD40 and SD0 with pomalidomide, but with PT179, only SD40 gets recruited. And also the KD values that we observe with TR-FRET are essentially the same as that we observe with BLI. So now, having established that the SD40 works as a Degron in cells and the in vitro properties are exactly what one would expect as would be expected, we asked ourselves, we still didn't really understand what the evolution did. So to understand that, we turned to structural biology, and we solved this structure of PT179 bound cereblon in complex with DDB1, that's its adapter, and these are all bound to SD40. And Moritz Hunkler, who's a cryo-EM expert in our lab, helped a lot with this structure. So the first thing we see is that SD40 is binding in a similar mode as any other zinc finger that we've seen binding cereblon before. But then this portion is very new. This is the C-terminal tail of SD40, where we see clear side chain densities. To see if there's a difference between PT179 and pomalidomide, we also solved a structure with pomalidomide bound to SD40, and we see that the structures essentially overlap, and hence we think that SD40 binds cereblon in an IMiD agnostic manner. It's IMiD dependent, but it's agnostic to which IMiD it is. Looking more closely at some of these mutations, the first question is, how was SD40 able to tolerate the bump? One of the early mutations happens at this position 18, where it's a glutamine to phenylalanine mutation. And we think that the phenylalanine is able to pack against this electron-rich morpholine ring, which was the bump, and hence it's able to tolerate this bump. Another early mutation happened just 2 residues down in the main zinc finger at position 20. And here, this glutamate changed to proline. When we look at what this proline does, it seems to be forming this constellation of hydrophobic residues and this kind of packing with 3 residues that are not mutations, 49, 45, and 36. This leads to this small hydrophobic pocket, which also packs against cereblon. And by doing that, it's stabilizing the C-terminal tail in this form because it's essentially letting the tail pack back onto the main zinc finger itself. And by allowing that, what happens is that this tail spreads across the N-terminal domain and forms many, many different contacts. And this increases the Barrett surface area from just the zinc finger portion being at about 1100 angstrom squares to 2300 angstrom squares. So it doubles the contact area and also has very high shape complementarity. So SC equal to 1 is what we say is perfectly complementary surfaces, and 0.68 is a very high complementarity. Some of these later mutations like 47 and 50, we think this contributes to the affinity of SD40 to cereblon. Specifically in the case of 47, which arose between SD8 and SD31, we see how the circuit activation jumps up. And this happens because 47 forms this pi-pi stacking interaction with this histidine and the N-terminus of cereblon, while also positioning the residue next to it, which is not a mutation, to have this pi stacking interaction with the C-terminal domain of cereblon, thereby essentially stapling cereblon into this closed conformation. And Y50 also has a similar role. So the next question was, could we use MG pace to evolve SD36 into something that also tolerates a different kind of bump?
Yeah, and so as many of you people in the audience probably know, mouse cereblon contains a valine to isoleucine mutation, and this ultimately prevents adequate zinc finger neosubstrate recruitment in mouse cells following administration of IMiDs. And so we figured, why don't we try implementing MG pace in order to rescue these unfavorable contacts instead of at the molecule and protein interface, at the protein and protein interface within the ternary complex. And this would also potentially establish a zinc finger Degron tag that could be compatible in mouse systems with these compounds. And so initially we just turned to our transcriptional activation assay with SD0 and SD36 and just dosed in PT179. And surely enough, we could see that SD36 had some ability to bind to PT179 complex cereblon, at least at high concentrations. So we then initiated evolution and performed roughly 300 hours of pace, whereby we identified SD56, which contains 3 additional mutations on top of the SD36 genotype. We then turned back to that flow-based reporter assay, this time in mouse 3T3 cells, and you can see that following evolution, MG pace did improve the potency of this Degron by roughly 5-fold, as you can see between SD56 and SD36. And so we have these 2 Degrons, one being one that's compatible with human systems, SD40, and one that's compatible with with mouse systems, SD56. These are also readily degraded by canonical IMiDs, and we're just showing a subset of those data with pomalidomide down here, where you can see SD40 in human cells and 3T3 cells and mouse cells are degraded at nanomolar concentrations of pomalidomide. And we've also tested other canonical IMiDs such as lenalidomide and thalidomide with SD40, and we see a similar trend.
While we were evolving these Degrons, a very interesting paper came out, which talked about how these IMiDs bind cereblon and change it from this open structure to this closed state. In particular, mezigdomide was found to bind cereblon both at the N-terminal end and at the C-terminal canonical IMiD binding pocket, and also at the N-terminal side. And if we compare this to our structure, we see SD40 does something very similar. It binds the C-terminal as well as the N-terminal end. So what happens when we use mezigdomide with SD40? Well, what happens is that we get single-digit picomolar degradation in a matter of hours, and this is nearly complete degradation of SD40 tag GFP. So what we think happens is that mezigdomide already staples cereblon shut, and hence SD40 is able to then go and bind more tightly. And the same effect also happens in mouse cells with SD56.
And so just to conclude, we have basically presented this clean and selective IMiD derivative, PT179. We've also described this new directed evolution system called MG pace, which enables the continuous evolution of molecular glue interactions. And we applied this system to evolve SD0 with these morpholine substituted compounds. This process identified ST40, which is a compact 36 amino acid Degron, which responds to nanomolar concentrations of PT179, canonical imids, and also next generation imids such as mezigdomide. An ST40, given its small size, is efficiently knocked in to endogenous loci using prime editing, which enables pretty facile evaluation of endogenous protein knockdown. Also, as Shourya presented, we've elucidated this beautiful cryo-EM structure of the evolved ternary complex, which not only reveals new contacts with the N-terminal domain of cereblon, but also an extensive interface with both the thalidomide binding domain and the N-terminal domain. And these kind of extensive contacts seemingly promote closure of cereblon and lock it into that putatively active conformation. We also applied MG-PACE to evolve a mouse, cereblon-compatible Degron ST56, and this Degron tag also responds to PT179 and canonical imids in mouse cells. And so with that, we'll go to acknowledgements, the most important slide. First, I'd like to thank Professor David Liu for his support and guidance throughout this entire project. He's a fantastic PI, and it's a great lab to be in. I'd also like to thank Jaron Mercer, huge shout out for initially serving as my mentor and really just teaching me the ropes of PACE and everything in the lab. He's been a fantastic partner, and it's been a really great time, not only learning from him, but also learning with him throughout this entire process. I'd also like to thank Drew Nelson, who helped with some of the prime editing experiments that we performed, and also Peter Chen, who helped with some of the RNA-seq analysis.
And also thank you to Eric Fisher and Amit Chaudhary, who also guided us throughout this project and gave us some key inputs. And Moritz helped a lot with the cryo-EM structure, and Catherine did a lot of the proteomics that was essential for this paper. And thanks, of course, to both our labs, the Liu and the Fisher lab, and these are our funding sources. Thank you for listening.